Good morning, children. In this video, I'm going to talk about C4 cycle, and this topic is there from chapter 13, Photosynthesis and Higher Plant, from class 11th Biology. So let's begin with it. C4 cycle is also called Hatch-Slake cycle. It is based on the name of scientists who have thoroughly studied this particular cycle. Let's go ahead with the introduction of it. What is the C4 cycle? In biosynthetic phase of photosynthesis, where we see fixation of carbon dioxide, C4 cycle is one of an alternative and a very efficient pathway by which plants are fixing carbon dioxide. So other than C4 cycle, majority of the plants, they are fixing carbon dioxide with the help of C3 cycle. But there are certain plants which, for example, like maize and sugarcane, they are using an additional pathway, and this pathway is called C4 cycle. So why this is called C4? Because the first stable product of this pathway is a C4 acid, and that is oxaloacetic acid. Due to the presence of this acid, it is called C4 pathway. And C4 acid, it donates one carbon dioxide to the C3 cycle. What does that mean? That means the plants which carry out C4 cycle, they also carry out C3 cycle for the formation of glucose, isn't it? So in addition, they have the C4 cycle. So who provide carbon dioxide to C3 cycle here in this case? It is the C4 acid which is being formed in C4 cycle. So what happened until mid 60s? It was believed that only the C3 cycle or the Kelvin cycle is taking place in the plants for the fixation of carbon dioxide. But later in 1957, Kortchak and co-workers, they reported a synthesis of 4 carbon organic acid as the first stable product in sugarcane. And the same was reported in the year 1960 by Karpilov. And later on in the year 1966, two scientists, as I told you, Hatch and Slake, they thoroughly investigated this and they came up with this pathway, and they confirmed the presence of C4 cycle in certain plants. So we have discussed about all these things already. Okay, so that's about the background of this particular cycle. Let's go ahead with the features of C4 plant. So first of all, these plants are found in tropical and subtropical region. These plants, they are showing Kranz anatomy. Okay, what is the meaning of Kranz anatomy? That the vascular bundles are surrounded by layer of bundle sheath cells having large number of chloroplast. So here the mesophyll cells and bundle sheath cells both are participating in C4 cycle. Next thing, that the chloroplast which is present in C4 plants are dimorphic in nature. What is the meaning of dimorphic nature? That means they have two morphologically distinct types of chloroplast. First one, or first variety of chloroplast, is present in bundle sheath of cells which are larger in size and arranged centripetally. They have starch grains but they lack grana. But when we talk about mesophyll cells, there the chloroplasts are normal, as we have seen in other plants. So this is about features of C4 plants. Let's talk about its biochemical pathway. This is a diagrammatic representation of C4 plant, wherein you can see that how mesophyll cells are participating, and other than mesophyll cells, bundle sheath cells are also participating, and the cells are connected by plasmodesmata. Now what happens here? The carbon dioxide is fixed in mesophyll cells and it is converted into C4 acid. This C4 acid is transported to bundle sheath cells, where it releases one carbon dioxide or gets decarboxylated, and this CO2 is utilized by C3 cell. C3 cycle, and then the C3 acid which is formed now will be transported back to the mesophyll cells for regeneration of an enzyme. So let's look at this biochemical pathway in little detail. So let's go ahead with the steps how this particular pathway is taking place one by one. So what happens? Carbon dioxide from the atmosphere, it enters into the mesophyll cells as we have seen, and it combines with phosphoenol pyruvate, which is a C3 compound, and it forms a 4 carbon compound which is called oxaloacetic acid in the presence of an enzyme called phosphoenol pyruvate carboxylase. Fine, as you can see, so PEP, which is the acceptor of carbon dioxide, when it is getting converted into oxaloacetic acid and phosphoric acid. Now this oxaloacetic acid is converted to malic acid and in the presence of a malic dehydrogenase enzyme. Fine. So this is what basically happens. Now the C4 acid which has been formed, it is transported to bundle sheath cell. It could be malic acid, it could be aspartic acid. So this C4 acid will be transported into the bundle sheath cell. For example, malic acid. Now this malic acid will be decarboxylated in the bundle sheath cell, okay, and it becomes pyruvate or gets converted into pyruvate. The carbon dioxide which is released by malic acid will be taken up or will be utilized in C3 cycle. Did you understand? So this is something which is called, which is basically happening here. So malic acid would be converted to pyruvate and it releases one carbon dioxide. This carbon dioxide will be taken up by the C3 cycle which is taking place in bundle sheath cell, and it will be utilized for the formation of carbohydrates or sugar. The pyruvate thus formed by decarboxylation, it returns to the mesophyll cells where it will regenerate phosphoenol pyruvate, that is PEP, in the presence of an enzyme pyruvate phosphate dikinase. So this is what the reaction is all about. So that's about the biochemical pathway of C4 cycle, and let's talk about its significance. So why the C4 cycle is important? The plants which carry out C4 cycle, they live in area with high temperature. And when we talk about these plants, they can tolerate very high saline conditions, and even if they have high temperature, even if they have higher saline conditions, still they are able to fix carbon dioxide. Next thing is high carbon dioxide fixation. If we compare these plants with the C3 plants, what we notice that they fix higher carbon dioxide or the carbon dioxide fixation is higher in their case. The rate of transpiration is very less because they are able to carry out photosynthesis even if the stomata is very less open or almost closed. In that condition as well, they will be able to carry out photosynthesis. Photorespiration, which is a wasteful process, is absent in case of C4 cycle. And as I told you that they can carry out photosynthesis even if the stomata is almost closed. So that's all about C4 cycle. Thank you.